In glycolysis, reactions 1, 3, and 10 all have large free energy changes. So these are the regulated reactions. The activity of the enzymes that catalyze these reactions is either increased or decreased under specific conditions. Let's start with reaction 1. Reaction 1 is catalyzed by the enzyme hexokinase. The activity of hexokinase is inhibited by glucose 6-phosphate. Since glucose 6-phosphate is also a product of the reaction, this is an example of feedback inhibition. Feedback inhibition is the inhibition of an enzyme by a reaction product. As more glucose 6-phosphate is produced, the reaction rate slows down. Hexokinase governs the rate-limiting step of glycolysis in the brain and in red blood cells. In most cells, glycolysis is regulated during reaction 3. This is important whenever the rate-limiting hexokinase reaction is bypassed, such as in the breakdown of glycogen, which leads to the formation of glucose 6-phosphate rather than glucose. Like reaction 1, reaction 3 results in a large negative free energy change and ATP is consumed. Reaction 3 commits the cell to completing the glycolytic pathway. It acts as the valve that controls the flow of substrate through the remaining steps. Reaction 3 is catalyzed by the enzyme phosphofructokinase, or PFK. Both activators and inhibitors regulate PFK. Activators increase the enzyme's activity, whereas inhibitors decrease its activity. In glycolysis, ATP inhibits PFK activity. This makes sense, since the purpose of metabolizing glucose is to generate ATP. If enough ATP is already present, there is no need to make more. The product of reaction 9, PEP, also inhibits PFK activity. Even though these molecules aren't the direct products of reaction 3, they are both examples of feedback inhibition. Activators of PFK include ADP and AMP. Their levels increase when ATP levels are low, thus reversing the inhibitory effects of ATP. Citrate also affects PFK activity. Citrate is part of the TCA cycle. Predict how citrate affects PFK activity. Is citrate an inhibitor or activator of PFK? Enter your answer and click Submit. Yes, that's correct. No, that's incorrect. Citrate inhibits PFK activity. This is another example of feedback inhibition since citrate is part of the TCA cycle. High concentrations of citrate indicate that enough metabolites, such as pyruvate, are already feeding into the TCA cycle, and there is no need to make more. The regulation of PFK activity by citrate links glycolysis to the TCA cycle. The enzyme pyruvate kinase, or PK, catalyzes reaction 10 of glycolysis, the step that forms pyruvate and generates ATP. The action of this enzyme results in a large negative free energy change, so its activity may be regulated. Reaction 10 commits the cells to either anaerobic fermentation or the TCA cycle. Like the enzyme of reaction 3, many different effectors regulate PK's activity. The steps that regulate glycolysis reactions affect the concentration of glucose in the blood. Blood glucose levels decrease as the production of ATP increases. When ATP production slows down, less glucose is consumed. In addition, insulin and other hormones and regulatory systems tightly control glucose levels. If glucose levels are too high, excess glucose is polymerized and stored as glycogen in animals or starch in plants. If levels are too low, glucose is released from these molecules and made available for use by cells. The regulation of glycolysis illustrates the general principles that apply to the regulation of all biochemical pathways. Glycolysis takes place in the cytoplasm. It consists of 10 reactions that convert glucose to pyruvate. These reactions can be divided into 2 phases. The first phase is the investment phase, and ATP is consumed. The second phase is the payoff phase, and ATP is generated. There is a net generation of ATP. 2 ATP molecules are generated for every glucose molecule entering glycolysis. In addition to ATP, glycolysis generates 2 molecules of pyruvate and 2 molecules of the reduced coenzyme NADH. Under anaerobic conditions, pyruvate is reduced to lactate or ethanol to regenerate the oxidized form of NAD. When oxygen is present, pyruvate is converted to acetyl-CoA, which enters the TCA cycle. NADH enters the electron transport chain, and more ATP is produced. 3 reactions of glycolysis are regulated. They are reactions 1, 3, and 10. These reactions all have large negative free energy changes. A large free energy change ensures that the reaction is essentially irreversible. Enzyme regulation is possible only when the free energy changes are large. Like glycolysis, all metabolic pathways are regulated. Regulation of metabolic pathways allows organisms to adapt to changing environmental conditions. For example, an animal might need a burst of energy to run and escape from a predator. Without control of metabolic pathways, energy could not be used efficiently, and the organism would not survive. Regulation takes place on many levels within the steps of each pathway, as well as among pathways, cells, tissues, and the organism as a whole.